Welcome so much to the symposium. We're honoring our 10 researchers who are part of our research apprenticeship program, UNLAB, or Understanding Nature in Los Angeles Biodiversity. We're going to hear each researcher talk about what they did over this last year. They developed hypotheses, they asked questions, they used many different methods. They analyzed data, they learned computer programming, they went out in the field. It's really a very impressive to see the work that they've done. So the way we're going to do this is we're going to start with 5 of the researchers. We're then going to take a quick 10-minute intermission, and we're going to have our next 5 researchers after that. So without further ado, I'm going to introduce our first researcher, Melissa Gonzalez, who's going to talk about thermoregulatory impacts of black carbon in songbirds of Southern California.
Thanks, Allison, for the introduction. Hello, everyone. My name is Melissa, and I'm one of the UNLAB researchers here at the museum. And I've had the pleasure of working with Allison in the ornithology collection, doing research on songbirds. And before I get started, I just wanted to thank everyone who was involved in making this program happen. It has made such an impact in my life, and it truly made me envision myself as a scientist. So I just want to say thank you from the bottom of my heart to all that were involved for your efforts and for caring about creating an inclusive community for future generations in science and research. And now I'll talk about what I've been doing the past year. So in recent times, here in Southern California, air pollution has increased due to having more motor vehicles, industrial factories, and wildfires, which all contribute to letting out fossil fuels. They all let out many harmful chemicals, but specifically black carbon. And black carbon comes from fossil fuels and from biofuels, which we all produce. One thing in particular that makes black carbon a concern is that it absorbs heat or sunlight, but we aren't sure the impact it may have. And specifically, birds have shown to be very sensitive in air pollution. We know that it affects their health in various ways, but we don't know the impacts that black carbon may have on their feathers. One thing that we do know is that feathers can capture black carbon in the air. There was a study done by Shane Dubé and Carl Foldner where they went through museum bird specimens in the Chicago Museum and found that they had black carbon on their feathers. So they found that birds from earlier time periods had more black carbon on their feathers. Yeah. Due to historical changes in coal burning and environmental protections that have reduced air pollution. And recent work in our own collection has shown similar patterns in LA. In LA, air quality has gotten better overall, but due to heavy reliance on motor vehicles and the frequent wildfires in Southern California, bird specimens still show black carbon on their feathers. So, however, no one has really studied what potential impacts black carbon may have on feathers for birds. And as we know, feathers are very important for birds. They have many functions. Specifically, they help with thermoregulation. And thermoregulation is when feathers help keep a stable internal body temperature, which is crucial for bird survival. And one way birds are able to do this is through absorbing or reflecting near-infrared radiation, which is solar radiation. Here I have an electromagnetic spectrum, which shows all the different types of wavelengths. Us humans are only able to see visible wavelengths, which we experience by seeing colors. But near-infrared radiation, or NIR for short, are wavelengths longer than visible light, and we can't see NIR, but we can feel it when the sun warms up our skin. And birds experience the sun warming them up too, except they have feathers to protect their body. Feathers are able to reflect NIR radiation to help reduce heat. However, we know that black carbon absorbs NIR radiation, so if there's more black carbon on bird feathers, it raises the question, does it impact their ability to reflect NIR radiation properly? So knowing that there has been more black carbon in the air recently, and it is able to stay on feathers even decades later, I was interested in knowing how does black carbon affect NIR reflectance using time period as a proxy. So I wanted to test and compare NIR reflectance in bird specimens from different time periods from LA. I wanted to know factors like species, era, sex, and body regions could cause variation in results. I specifically chose to test whether sex has an impact because different sexes have different behaviors, and for body regions, they have different functional differences for a bird. So I used museum specimens just as Dubé and Foldner. I started off by sampling 6 different LA resident songbird species here in our own collection. I sampled the American crow, the California scrub jay, the northern mockingbird, black phoebe, house finch, and the lesser goldfinch. I sampled 10 males and 10 females from each species from 2 different time periods, historic and recent. The historic period includes birds from the 1880s to 1935, and the recent period includes birds from 1970s to present time. And I sampled about 360 birds in total. I measured 3 different body regions on each specimen for all 6 species, which were the throat, the belly, and the mantle. I chose these regions because the throat is important for display when courting a mate, and the throat for insulation, and the mantle being that is the most exposed to the sun. I also used a device called a near-infrared spectrometer that measures near-infrared wavelengths at all angles when pointing a probe at each bird. And here is an example of how house finches from historic and recent time periods visibly look. So the historic specimens, which are on the left, visibly have more black carbon and look dirtier and darker, whereas the recent specimens on the right are lighter and cleaner. But it's not sure if these visible differences are the same in the near-infrared spectrum. So when I tested the different factors, results showed that species, era, and body regions were all significant, but sex was not. And for the sake of time, I'll only walk through what results I saw on body regions across species. So when I measured NIR reflectance in the throat, belly, and mantle, I saw that the belly, which is highlighted in blue, shows consistent differences through time. However, one species, the lesser goldfinch, its other body regions also showed higher NIR reflectance in recent times, which is boxed in black. However, in the American crow, all of the body regions show lower reflectivity in recent times, which is boxed in white. So one thing I noticed about regions that were significantly different is that they are typically visibly light or white. And in the case of the lesser goldfinch, all of the significant regions were brightly colored. So this suggests that it's light or bright colored regions that are most impacted by black carbon. In the American crow, we expected to see lower NIR reflectance in historic times due to more black carbon exposure because of coal burning in the past. But I saw recent specimens had lower NIR reflectance instead. We noticed that historic specimens look more faded out compared to recent specimens, and we think that it may be due to melanin pigmentation fading over time and making the bird lighter and reflect more NIR. But we still need to test this further if this is true or not. So in conclusion, we believe that most important differences in NIR reflectance is related to feather color, and overall it does seem that black carbon does have implications for thermoregulation in birds. And I want to thank NSF for funding this research, even if its funding did get cut off in the end. I was still able to complete this project and also be part of many different experiences. And I want to thank the staff at NHM. I appreciate all the support and guidance that I received along the way. And I want to thank my UNLAB cohort. I'm happy to have met such a cool and diverse group of people to share this experience with. And on a last note, if you're interested in participating in bird observations to investigate bird responses towards air pollution in LA or California, you can join Project Phoenix, which is a community science project that Dr. Allison Schultz and others are working on here at the museum. So anyone's welcome to join, and you can find more info on the website using this QR code. Thank you.
And I think we have time for a couple of questions. Any questions for Melissa?
Just raise your hand and we'll come over with the... Great talk, Melissa. A question regarding the feather stuff with how the carbon is accumulating. This is an odd question to ask, but do you think there are any behaviors that the birds can do to get that carbon off by chance? Like, I know it's sometimes, like, say, for example, like Dusty, like taking a bath. Can birds do anything behaviorally to get the carbon off, or no?
I'm not entirely sure. I just know that they do take baths and dust baths, but other than that, I'm not sure. But thank you for your question.
Once again, thank you so much. That was an amazing talk. I was wondering why you chose these two different time periods, especially for the recent specimens. Were they simply the specimens that you had on hand, or did you specifically choose that time range for a reason?
I chose those two different time periods because in the historic period, there was more coal burning, so we know that birds were dirtier. And as of now, like the recent specimens, since there's more environmental protections in place, they're more cleaner supposedly. So, and the type of songbirds that we used those because those were the ones that were available in our collection, and also because they're resident songbirds, which means they're here all year long.
Thank you so much, Melissa. Great job. Our next talk is going to be by Jasmine Gallardo, who's going to talk about temporal trends in microplastic ingestion by Southern California opali, 1940s to present. Thank you, Jasmine.
Okay. Thank you for that introduction. So again, my name is Jasmine Gallardo, and I am the UNLAB researcher in the ichthyology, or the fishes department. And before I begin, I wanted to pose a question for everybody in the audience. So can you please raise your hand if you think that you might have ever ingested or eaten a microplastic? Nice, okay. And I'm pretty sure I saw everybody's hand raised, but if that wasn't the case, I just want to let everybody know that it's my opinion that we should all be raising our hand, and I'm going to talk a little bit about why that might be the case. So before we dive deeper into my research, I'd like to begin by defining what exactly a plastic is. So plastics are synthetic petroleum-based materials and can be made into multiple shapes, sizes, and colors. So on the screen, we have a few different examples of rather large plastics. On the other hand, microplastics are pieces of plastic that are 5 millimeters or smaller, with different types including fibers, fragments, films, and pellets. A microplastic can be made into these sizes straight from the factory, as seen with the image on the right, or they can be broken down from larger plastics over time. Regarding the history of plastics, it wasn't actually until 1907 that the first fully synthetic plastic, bakelite, was produced. For the next 30 years or so, many popular plastic types were created. However, it wasn't until the onset of World War II that plastic manufacturing really began to skyrocket. At this time, plastic was used for wartime activities, but it also became commercially important for everyday consumers when materials such as metal and wood were being rationed for war efforts. In 1972, the first instance of small plastic debris was first documented in the marine environment. But it wasn't actually until 2004 that the term microplastics was first properly coined, meaning it's a relatively new field of research in terms of the larger scheme of science. Now, you might be asking yourselves, where are microplastics found? And the truth is that they are everywhere that we can think of, in the air, in the water, and in animals, including us as humans. Most of these data are based on modern studies, which at first makes sense, since we don't have time machines to go into the past and figure out how pollution might have impacted us. However, with natural history collections, such as those housed here at the Natural History Museum of Los Angeles, we can use preserved specimens to travel back through time and discover more about the world that past generations lived in. There are very few temporal studies of microplastic ingestion by fishes, and even fewer in marine systems. So therefore, that brings us to the study that I am part of, where my lab is using historical samples of 6 different marine fishes found all throughout the Southern California coast in order to determine impacts of plastic pollution across time. These species include the northern lampfish, northern anchovy, Pacific viperfish, California halibut, and shiner perch. Here we have a mix of deep water and more shallow water plankton eaters, invertebrate eaters, and fish eaters. However, you might have noticed that there is one group on the food web that we are missing with these animals. And that brings us to my study species, Scarlet nigricans, or the opali. So when opali are young, they tend to reside within tide pools where they begin to eat various types of algae before heading into the broader ocean once they're about 20 centimeters in length. The opali is actually one of the only herbivorous fish in all of Southern California, and it's very well represented in natural history collections. The two main questions that we wanted to answer with this study species are, has opali plastic ingestion changed across time? And what types of plastic are being ingested by opali? In order to answer these questions, I would remove the entire gut from a preserved specimen before chemically digesting it. I would run a particle separation, vacuum filter gut contents onto a glass filter, and use a Raman spectrometer and its laser to determine whether or not plastics were being ingested by the fish. So far, we have dissected 48 specimens and analyzed 12 of them. Of our 12 analyzed samples, the oldest was from 1949, and the most recent was collected in 2011. Our preliminary results show that plastics have been found in every decade analyzed so far, with plastic particles being found in opali before any of our other study species. To put it into context, the second oldest sample to have microplastics was a northern lampfish specimen from 1957, meaning there's actually an 8-year difference between when the opali ingested plastics versus any of the other species in our study. We have also found that opali have ingested a much wider variety of plastic types compared to any of the other species that we've studied from Southern California so far. This is interesting compared to the 3 to 5 plastic types that have been found in each of the other species so far, because with only a sample of 13 guts analyzed, we've identified 7. Pretty interesting finding for us. We hypothesize that the historic presence as well as the variety of plastic in opali might be because of the opali specimens used for this project being collected around the Palos Verdes Peninsula. For context, this means that many of the opali actually lived in and around the Port of Los Angeles, Fort MacArthur, and many popular tide pooling areas. The Port of Los Angeles is the busiest container port in all of North America, not just the United States or California, and it opened in 1907. It is situated in the Los Angeles harbor, where many polluted channels lead into waters that are eventually inhabited by the opali. In addition, plastic-containing ships and machinery are also stationed in and frequently enter and exit the port as well, potentially contributing to the historic ingestion of microplastics by opali. In addition, Fort MacArthur is now a retired military fort in San Pedro, but it was active during World War II, and the use of various types of plastic in wartime activities through bombs, parachutes, and more could potentially contribute to the early presence of plastic in opali. The constant presence of humans for various leisure and research activities in tide pools also can introduce a range of plastic types through litter, towels, bathing suits, fieldwork materials, and more. The small pools of water also allow for a lot of agitation and sun exposure, which can further break down large plastic pieces into easily ingested microplastics. Now, why does this research matter? The One Health approach urges us to recognize the interconnectedness between humans, animals, and the environment. We now have an understanding that plastic pollution might have been impacting opali decades before we even knew that microplastics existed. This can pose environmental risks for the marine environment, fisheries, and other commercially important organisms. In addition, understanding more about how microplastics have negatively impacted the health of fishes can give insights into how they can impact humans, with previous studies in fish showing microplastics linked to poor reproduction, poor immunity, nutrient absorption, and in some cases, even death. By establishing the historical presence of microplastics in Southern California marine waters and beyond, we can begin to piece together how these microplastics may have affected us in the past and work towards a healthier future for the environment, animals, and ourselves. Therefore, in the future, our lab hopes to complete sampling and analyses of the opali specimens, cross-examine our findings with other Southern California species, and expand our work into Northern California to create a more holistic view of our coast history with plastic pollution. With that, I would like to thank these communities, individuals, and institutions, as well as my funding sources, the California Ocean Protection Council, California Sea Grant, and National Science Foundation. I would also like to acknowledge that this is an institution of, for, and with Los Angeles. And like the city, I could not and would not be here today without the contributions of the queer, immigrant, and Mexican communities. Thank you.
Great job, Jasmine. So now we have a few minutes for questions.
And just a note, if you'd like to ask a question in Spanish, we are also happy to have that.
Fantastic talk, Jasmine. It's just a what-if question. Do you think if you had specimens, say, along the Central Coast where, you're away from the port, do you think there'd be less plastic across that time span?
I think there would likely be multiple factors contributing to this. So one thing that we're finding with the opali is maybe not necessarily how close that they are to the port, but it could just be in general that they're closer to the coastline than some of the deeper water species. But there could also be things such as when we're looking at our invertebrates or the invertebrate eaters versus piscivores, the fish eaters, we have differences there. So it really depends. There could also be factories near the Central Coast that could be impacting things out there, other types of military activity or travel, things of that nature. So I can't really guess. The world of microplastics is really broad.
Thank you.
We have one more question.
Thank you once again for bringing so much attention to microplastics. I have a little bit of background in microplastics, and in my research, fish consumed black plastics because it resembled food. So I'm wondering if the fish you studied were attracted more to red and green algae because that was something in their normal food cycle and stuff like that.
Yeah, so one thing about the way in which we approach the analyses of our plastics, I kind of went over it for the sake of time, but we use Raman spectroscopy without having to visually identify any plastics ourselves. So we're relying on a random 1 centimeter square area scan of the filter. So we aren't at this point identifying types or colors of the plastic beyond what the chemical signature is for them. So I don't know if colors would necessarily have any impact on it. My assumption at this point would have to be with the way that opali in particular eat, they are sucking in food. So it could even be they aren't recognizing plastics at all. They just happen to be ingesting it. They also often ingest sand and other invertebrates on accident, just with the way that they eat. So it could be multiple factors. Thank you.
Thank you so much, Jasmine. For our next talk, we have Adrian Garcia Sanchez, who will be presenting Conjuntos de Vertebrados Marinos de la Formación Capistrano del Condado de Orange. Thank you, Adrian. This talk will be in Spanish.
Wait, up.
Bienvenidos al Museo de Ciencias Naturales. Mi nombre es Adrián García Sánchez y gracias Alison por esa introducción. Y sí, mi proyecto es en conjuntos de vertebrados marinos de la formación Capistrano y trabajé con Jorge Vélez Forabe, curador de mamíferos marinos en el vertebrado de paleontología aquí en las ciencias naturales, y con James Parham, profesor de geología en la Universidad de Florida. Y como dice mi familia, ¿verdad? En la vida es más sabrosa. En el mar es más sabrosa, perdón. Pero es cierto que Los Ángeles estaba sumergido bajo durante los últimos 90 millones de años. Y aunque la Countdown, que es parte de la formación que estudié, no era parte de Los Ángeles, pero fue formada durante el mismo entorno similar. Y aquí podemos ver que esa sección de parte de Orange County fue donde la formación Capistrano fue formada. Y fue formada de 15 a 20 millones de años. Y vamos a ver ahorita que las investigaciones previas de Michelle M. Barbosa, que ella ayudó a mantener una edad de esa formación usando los dientes de un caballo, y esta dio luz a una de las unidades que está encontrada en esta formación. Y vamos a ver que hay dos unidades, hay un oso y un silt. Oso es como más de la costa, arena más coarsa, y el silt es una arena más fina. Y están pensando, ¿qué es una formación, verdad? Y una formación es una sección de unidades completas de piedras y sedimentos. Y con esta formación nos puede decir la historia que sucedió durante esta época. Y la formación fue formada y tiene una edad de 5 a 15 millones de años, obviamente con la ayuda de Michel Barbosa y sus estudios. Y también sabemos que fue depositada en un entorno marino, que sabemos que en el oso, que es en la parte más derecha, es una unidad que tiene parte de la costa, agua poco profunda, y también tenemos el silt layer, que sería el agua más profunda. Y sabemos que contienen los sedimentos marinos y no marinos. Y ahora, aquí es una litología de cómo se puede ver en la superficie. Obviamente es en Los Ángeles, pero es Orange County. Y abajo tenemos la formación Capistrano, arriba del Monterrey, y vamos a enfocarnos en esas dos formaciones para este proyecto. Y las preguntas que yo quería contestar con mi grupo sería, ¿qué nos puede decir la fauna fósil sobre la formación Capistrano? Y si existen diferencias de la fauna fósil entre las dos unidades dentro de la formación o con la formación Monterrey que está bien abajo. ¿Y qué nos puede decir la evidencia sobre el paleoambiente? Que es muy importante ahorita con todo lo que está pasando con las excepciones de muchos de los animales sobre la costa de Los Ángeles. Y empezamos con los métodos, ¿verdad? Y estudiando esta formación fue ir por la colección de estas unidades y examinar cada fósil y las especies usando un microscopio y una lectura publicada como Michel Barbosa, y vimos casi 2.000 a 2.500 huesos y especímenes completos. Y también después de eso construimos una lista de especies que se pudieron identificar y ayudar con la ayuda de mi mentor Jorge y Jen. Lo pudimos subir a RStudio, y con la ayuda de Allison también estudiando pudimos hacer una grafía que nos enseña cuántas especies había en el oso, que eran 42, y juntos teníamos 16, y en el silt teníamos 32. Y cuando no se puede identificar los registros, compararnos con los huesos de especies de ahora, usamos este método. Cuando estudiamos los morfolotipos y tomamos fotos de cada cráneo. En la mano izquierda tenemos un cráneo de la formación Capistrano de un delfín, y la mano derecha es uno de los cráneos de los delfines que se pueden encontrar en esta época ahora. Y comparamos y vemos si hay similares en los morfolotipos, y la verdad sí. Pero no nada más se usan los cráneos, para los delfines se usan los oídos del cráneo, pero el cráneo se mira más chido para esta presentación. Y como cada proyecto que vamos a ver ahorita, hay complicaciones, ¿verdad? Y con todos los proyectos, obviamente, hay complicaciones, y en las colecciones en este edificio necesitaban actualización, ¿verdad? Pero con la ayuda de Jorge y los del departamento, pudimos avanzar con el proyecto y examinar los conjuntos vertebrados de cada formación. Pero sí tomó mucho tiempo. Pero ahora vamos a los resultados. Y empezamos con los resultados, y no nomás examinaron los fósiles, pero también dónde fueron encontrados. Y aquí tenemos unas imágenes del mapa de la condado de Orange County. Los círculos de café, más bien anaranjados, amarillos, en la parte norte son del unidad oso, que tienen la arena más coarsa, ¿verdad? Y los diamantes azules que están en el sur eran de la unidad silty, que es la arena más fina. Y si se pueden ver, tenemos una sección separada, ¿verdad? Y así nos puede ayudar que sí tenemos una costa y un agua bien profunda. Tenemos dos separaciones, y eso nos puede ayudar a contestar las preguntas. Y ahora, cada unidad tenía su propia especie y obviamente sus especies que viven en su hábitat. Y empezamos con el oso, que tenemos el Pontolis cojona, que es una morsa. También tenemos un oído de una ballena Mississippi, y el cráneo de una sirena, y de un oído del delfín. Y del silt se puede buscar muchas de las especies que se pueden encontrar en esta unidad. Son como tiburones de mar profundo y ballenas, pero los más interesantes fueron este cráneo de un ave que se parece como un pingüino, que ya no existe obviamente en la costa de California, pero qué chido ver eso. Y también tenemos un cráneo de un talselión, que es una foca león, y de este fósil de un salmón que vivía antes en las costas de aquí de Los Ángeles. ese es mi lonche, ¿verdad? Perdón. Ese es el pescado que tenemos ahorita. Pero con todo esto, vamos a enfocarnos en los resultados entre la comparación de la Monterey y el Capistrán. Y aquí podemos ver que vamos a empezar con Mississippi, que son las ballenas sin dientes, y Odontoceti, que son ballenas con dientes o delfines. Y aquí en la mano izquierda, en anaranjado, sería la formación Monterey, y tenemos 4 especies de Mississippi y 9 especies de Odontoceti. Y en la formación Capistrano es lo verde que yo estudié con mi grupo, y tuvimos 3 especies y 9 especies. Y vamos a seguir con la familia de pinipero, o de morsas, y de focas, y de leones. Y empezamos con la mano izquierda otra vez, sería la formación Monterey, y tiene 9 especies que fueron encontrados completos. Y en la formación Capistrano, que otra vez nosotros estudiamos, fueron 11 especies. Un aumento, es algo para empezar ahorita. Y luego para terminar, las sirenas tenemos obviamente la mano izquierda de 4 especies en el contado en la formación Monterey y una especie en el Capistrano. Y ahora, volvemos a preguntarnos si podemos contestar cada pregunta de nuestro proyecto. Y primero fue, ¿qué nos puede decir la fauna fósil sobre la formación Capistrano? Y nos dijo que una biodiversidad significante estaba durante esta formación de morsas, y esta formación no se puede encontrar en otra formación, y tuvimos un aumento de 55%. Y la pregunta segunda fue, ¿existen diferencias en la fauna fósil entre las dos unidades? ¿Y qué nos puede decir de la formación comparado a otras formaciones? Y sí podemos ver una corriente especie que tienen similar ambientes en donde viven ahorita, y obviamente tenemos la diferencia no nomás en los sedimentos, que era la corza de la mano izquierda de oso y la arena más fina que será seal, pero comparado con las especies que tenemos ahorita en la costa, podemos ver que hay 2 significados y tienen sus propios ambientes que existen. Y eso nos trae a la última pregunta. ¿Qué nos puede decir la evidencia sobre el paleoambiente? Y obviamente con el aumento de morsas que tuvimos de 55%, también durante esta época sabemos que el kelp empezó a expandir. Y pensamos que por esa razón tuvimos un exceso de morsas durante esta época, y los bosques de kelp atraen a los pescaditos y a los animales, lo que sea, que los morsas pueden comer durante este tiempo, y una protección de predadores como los tiburones grandes de esta época. Y van a pensar, ¿por qué todo esto importa, verdad? Y examinando las colecciones en museos, como mis colegas que van a estar hablando, nos puede seguir con la evolución y extinción de los animales que tenemos ahorita, y también nos puede seguir con la evolución y extinción, y estudiar la biografía de paleotectónica. Y una investigación futura que me gustaría seguir con este proyecto, ojalá Dios, ¿verdad? Continuando con este proyecto, me gustaría mandar dientes de tiburón para obtener una edad de la edad de silt member, y como no se puede mandar dientes de caballo, obviamente que no existen dientes de caballo en el mar. Ya sé que ya vieron esta imagen, pero estos no cuentan. Pero vamos a mandar dientes de tiburón para agarrar una edad en la unidad de silt member. Y para aclarar esto, hay un mineral que se llama strontium que se puede encontrar en aguas saladas, y se puede cementar en los dientes de tiburón cuando se caen, y tienen la opción para encontrar la edad. Y para terminar, Y para terminar, llegamos al final. Nomás te quiero agradecer a todos de mi familia, a mi novia, a mis mentores y mis colegas de aquí de UNLAB. Y gracias al museo por prestar sus colecciones para este proyecto, y NSC por los fondos antes que se terminaron. Y ya que estamos aquí, le quiero pedir a todos gracias por escucharnos. Y ya sé que Los Ángeles ahorita no es un lugar bien... Los Ángeles es un lugar difícil ahorita, ¿verdad? Con todo lo que está pasando. Y con la administración y los agentes de ICE, pero unidos se puede con todo, ¿verdad? Y me gustaría darle gracias por estar aquí. Es un honor estar aquí enfrente de ustedes, y pues viva Latinos, ¿verdad? Gracias.
Muchas gracias, Adrian. Y if you have time for a couple of questions.
¿Preguntas?
Muchas gracias por tu presentation. So, quiero saber de ese programa, ¿qué fue la parte más favorita tuya y por qué?
La verdad, del programa todo fue genial, pero siempre tuve la emoción de estudiar fósiles de dinosaurios, pero me tocó de mamíferos y la verdad estaba genial. Me saqué estudiando muchas ballenas y es algo que no se puede seguir con otro trabajo, ¿verdad? Y era algo que me seguí conectado con los mamíferos marinos y era algo genial trabajando en una institución como esta de aquí con nosotros. Gracias por esa pregunta. Yeah.
Okay.
Yes, yeah.
Gracias, Adriana. So our next talk will be by Caleb Eubanks, who will present on Can a Fragmented Past Be Trusted? Assessing Skeletal Representation Bias in the Fossil Record of Birds with a focus on the La Brea tar pits. Thank you, Caleb.
All right, so the La Brea Tar Pits is an active paleontological site near Koreatown, just about 20 minutes that away. So pretty nearby. If you haven't been there, please go there. For thousands of years, asphalt has seeped up through the Earth's crust, forming the famous tar pits, and I'll explain the quotation marks in a second. The La Brea Tar Pits is a unique window into the late Pleistocene, also known as the Ice Age. The incredible amount and diversity of specimens to be found have researchers and visitors alike coming from around the world to visit the site. You may be most familiar with this kind of image or of a huge animal getting stuck in the lake of tar. However, most asphalt seeps actually looked like that. So it's a little easier to see how animals may have actually stumbled in and gotten trapped. So yeah, that famous lake pit is actually water that got pumped into an old excavation site. So first of all, what is asphalt? Tar? There's a difference. So asphalt is a petroleum-based product considered the lowest grade of crude oil. And it is formed under when an ancient marine plankton would be buried under sediment and intense pressure and converted into oil. Whereas tar is produced from processing and distilling organic materials such as coal or wood. It is also known as pitch, and it was also used by the indigenous folks that used to live here and still live here in order to make all sorts of stuff. So this crude oil would make its way through the Earth's crust through cracks and faults in order to make these seeps or puddles on the surface. Animals ranging from towering mammoths and giant sloths to tiny lizards and bugs became trapped in the sticky asphalt, becoming preserved for us to excavate to this day. When animals perished, their bones would remain in the asphalt, forming layers of jumbled up bones over time. These bones are carefully excavated, cleaned, identified, and sorted by a team of tar pit staff and volunteers. So as you can see, not every animal gets preserved, and not even every part of the animal gets preserved. This means that certain types of animals are more likely to fall into the asphalt trap, and then those will show up more in our museum collections than obviously those that didn't end up there. Thus, our understanding of those animals that got trapped or are more likely to get trapped will be better than those that did not get preserved. When large herbivores would get stuck, their cries of panic would attract the attention of hungry carnivores, many of which hunted in groups. The most commonly found mammal specimens found at the La Brea tar pits are the direwolves, followed by the saber-toothed cats, both of which have evidence of hunting in groups. So often for every one or two large herbivores that got stuck, there are multiple carnivores stuck there as well, thinking they have found an easy meal. This phenomenon is known as the carnivore trap theory. Yes, that will be on the test. All right, so mammoths, mastodons, giant sloths, all these are very big and cool, charismatic mammals. But is there someone we are forgetting? Birds. I think they're pretty cool. So birds are particularly tricky to study. If you remember that biased fossil record I mentioned, birds especially fall into this because their bones are quite fragile. And many birds are small, so their bones are more difficult to locate and extract from the blocks of hardened asphalt. So in this project, I set out to figure out what kinds of birds can be found in La Brea tar pits and what parts of the skeleton are more likely to get preserved. Using a specimen database from the La Brea tar pits collection, Excel, and a statistical program called R, I was able to analyze a little over 113,000 avian specimens. So first things first, what kinds of birds are more likely to get preserved? I warned you all there was a test. Here we go. So if you remember that carnivore trap theory I mentioned earlier, the first instinct might be if that is true, then carnivores are more likely to get found by us thousands of years later. And Yes, you are correct. A huge portion of the bird specimens surveyed were hawks, eagles, vultures, falcons, and owls. The most common individual species is the golden eagle, which is shown here. I also analyzed around 413,000 mammal specimens in order to compare those two data sets. And it's interesting that carnivore bias is even more stark in mammals, with nearly 83% of all mammal specimens found belonging to order Carnivora. And I definitely need to point out that one order Carnivora, as opposed to like 3 or 4 orders of birds of prey, and even within that order, it is mostly just 2 species, direwolves and saber-toothed cats, that make up that order Carnivora within the data set. And so then those two, direwolves and saber-toothed cats, so there's this huge bias towards pack hunters. So I wanted to compare that data set to modern stuff. Like if you look outside, do we see half birds of prey? So what kind of birds do we come? You probably saw some pigeons coming in today. House sparrows, house finches, American crows. If you were lucky, maybe you got to see a yellow-rumped warbler, or an Anna's hummingbird, Cooper's hawk. So these are all pretty common backyard birds. So this is obviously not an exhaustive list of all the birds you can find around here, but you can see that a lot of these are what are called songbirds or order passerines, and comparatively less birds of prey. Only 3.8% of the specimens I surveyed were songbirds. So there are more questions here. Were songbirds less likely to get stuck? Perhaps songbirds are pretty small, so maybe you don't find them as often in that asphalt. Also interesting to note, the most common fowl by far was the very large turkey, as opposed to like the quails, which are much smaller. So could body size be affecting our data set? You might also be wondering about that unknown section. 27% of these bones, someone just said, yep, that's a bird. And so those bones are either fragments or they weren't diagnostic, which means you don't know what kind of bird they came from. Or perhaps it could have been something less studied, so less easily identifiable. Perhaps some of those little bones are somewhere where the passerines are. So in order to assess whether bone size could be affecting the data set, I set out to find out what parts of the skeleton are more likely to get preserved. So here is a quick reference of bird bones. And then the vast majority of the bones surveyed were hind limb elements, so like legs. 40% of all those bones are found below the knee, so below the femur. So if you think about it, it makes sense that you would find a lot of toe bones, because birds and we have a lot of toes. But 15% of all the bones were tarsometatarsi, which are a fused foot bone, which each bird should only have 2 of. So that's pretty significant that 15% of the stuff that we found was stuff that you should only find 2 of per bird. So that's also, by the way, the tarsometatarsus, in case you just don't automatically know what that is. And so what could be causing this bias? So perhaps more robust or larger bones are more likely to be preserved, because the tarsometatarsus is those 3 fused bones. Or maybe we're finding more diagnostic bones. So the tarsometatarsus, I can tell you what kind of bird that is as opposed to a rib. Yeah. So if you break down the array of bones individually, there's definitely a bias for larger bones, such as limb bones, over things such as the very fragile skull. My favorite possible theory for this bias for hind limbs is that birds would get stuck feet first in the asphalt, and the top half was carried off by a scavenger, perhaps only leaving lower limbs. Quick summary, the ray tar pit is a very unique depositional environment compared to like caves or more traditional dinosaur type sedimentation stuff. Also, most of the avian specimens we found were birds of prey, and there's a bias towards lower limb elements. And if I may, this may be the most comprehensive survey of the La Brea tar pit's avian fossils since the 1930s. And so there's a lot more to learn. With all good science, there's more questions that come up. So are taxonomic biases driven by body size, ecology, or actual abundance? Do major vertebrate groups all exist the same sorts of biases between lizards, birds, mammals, all that kind of stuff? How do the biases compare to other depositional environments, other tar pits that exist, cave environments, etc.? Also comparing the La Brea tar pit specimens to the greater fossil record as a whole. So exactly how unique is La Brea tar pits? And then kind of like the big picture one is, how do these biases impact our phylogenetic data, our understanding of where birds come from? If we have mostly birds of prey, we're going to understand those a lot more than, say, songbirds and hummingbirds. Acknowledgements. Thank you to all the collections managers that have put up with me asking so many questions about how to become a collections manager, all the curators that have helped Shout out to my cat, who's listened to me run this talk a couple times, and to my cohort for moral support. Thank you.
Thank you so much, Caleb. So I think we have a couple of minutes for questions. Any questions for Caleb?
Thank you, Caleb. That was a great talk. My question is, seeing this bias in the birds, I was wondering if you would expect to see a similar bias in like smaller or medium-sized mammals as well, where you might find more of their hind limbs stuck?
That was definitely a thought, because a lot of the stuff we find is very big-bodied mammals, but there's definitely a lot of lagomorphs that I saw in the mammal data set, so I'd be very interested in comparing small, medium, large mammals to birds. Yeah.
Any other questions? Scott?
Repeated questions. I wanted to know what you wanted to do next. What do you want, if you had the opportunity to continue doing research, would you continue working with birds or would you move on to the next subject?
I already came into this program just enamored with specifically bird bones. So if I can work on birds the rest of my life, I'd be happy. So yeah, this is a good choice.
I just had a quick question about the stratification of the bird bones within the deposits. Is there like them being hollow? Is there a specific region of deposits that you find more bird bones?
I'm not super sure because most of the stuff that I worked on was database. So unless someone wrote it down, I won't be able to figure that out. But that's definitely another future directions, is that a lot of these specifically tell you which pit and what part of the pit they were found. So that'll be interesting to compare, because I do know some pits have like more of a particular species than others because they come from different time periods. So that'll be definitely interesting.
All right, I think that's all the time we have. Thank you so much, Caleb. Our next talk will be by Chloe Saldana, who will talk about monitoring the impact of the Portuguese Bend landslide on rocky intertidal habitats in Rancho Palos Verdes. Thank you so much, Chloe.
Thank you. Now, before I share my project, I want to introduce the Rocky Intertidal Zone. It is a dynamic coastal area that is constantly exposed to fluctuating tides. It is home to a wide variety of aquatic organisms that have adapted to the changing conditions of this environment, such as the anemones and sea stars pictured here. Now, to give some context of where my study site is located, here's a map showing Los Angeles. My study site, Rancho Palos Verdes, is a coastal city located in the southern part of the county. The city is known for its incredible view of the ocean, rough cliffs, and its rich natural habitats. Here's another view of the site. This area of the shoreline was impacted by the landslide, including these large isolated pools. Now, to give some context for the background of the landslide, it is of geological significance. It became active again in the 1950s, but showed little to no movement for several decades. But that changed a few years ago. I'm not sure if everyone here is local to Southern California, but in late 2023, we experienced heavy rains, which was really great for our plants and animals, but not for the landslide. The heavy rains, in a way, assisted in reactivating the landslide. The landslide forced locals to move out of their homes due to the irreversible damages it caused. But something that wasn't well reported was that the landslide was causing change in shape to the shoreline at a very rapid pace. The photo on the left illustrates the ongoing movement of the landslide moving downward towards the base of the hill. Some of the early reports indicated that the land had been shifting significantly, at times moving as much as a foot per day. The photo on the right shows the residential areas that are being impacted by the landslide. Near the top of the image is the Portuguese Bend Beach Club, which we passed each time we visited our site. A typical day out to the site consisted of a 5-mile hike. This is a cross-sectional view of the landslide. This shows that below the surface lies a clay-rich layer that has contributed to uplift due to its buoyant properties. As a result, areas that were once subtidal have been uplifted and are now exposed. This sudden change has led to a significant loss of the original subtidal communities that were there.
Now, to give some context of what the subtidal zone is, here's a chart that displays different tidal zones. Our subtidal zone, it means is remain continuously submerged underwater. For our project, we want to focus on a few different objectives, including documenting the changes that are happening to the coastline, monitoring the disturbances and recovery over time, and analyzing and describing the physical distribution spatially across Portuguese Bend. For our study site, we visited monthly. Our site consisted of 3 different sections, with each section having 4 to 5 fixed transect locations. This included our control site, which was minimally impacted by the landslide, our central, which had moderate impact from the landslide, and our eastern, which was heavily impacted by the landslide. To collect data, we utilized transects to survey the area. They were evenly spaced and run perpendicular to the shoreline. And along each transect, we used a photo quadrat to take high-resolution photos every 3 feet. This allowed us to cover more ground versus the traditional way where you identify and count species out in the field. We also collected data based on various physical attributes. But once we returned back to the museum, we uploaded our photos to an online platform called CoralNet. It is a tool used by researchers to analyze high-resolution images for studying reefs and coastal ecology. This platform helps classify biodiversity from each of our photos. And throughout the course of this project, I analyzed over 2,000 images in CoralNet. This visual representation shows just how dynamic the shoreline is. We can compare the shoreline of October 2023, before this land reactivated, and see that the exposed gray rocks are expanding. But by late 2024 and early 2025, we start to see even more dramatic changes, including of the formation of those isolated pools I pointed out previously. In the eastern section of our study site, changes in the coastline have led to the formation of a small bay. This was taken in December of 2024. Now, 5 months later, we observe a noticeable change in the shape of the bay. Here, this first photo was taken in October 2024. This was by one of our transects in our eastern section. This area was once inhabited by large sea slugs called sea hares. We now see in April of this year that the area is completely destroyed and replaced with eroded boulders, with sadly, no more sea hares. Also in our eastern section, we see constant erosion beginning to undercut the rocky shore, causing a natural bridge to form. This was taken in April of this year. And we see, less than a month later, it collapses due to constant wave exposure. So earlier I had mentioned the former subtidal communities being uplifted and exposed. Now I want to take the time to highlight some of that death and destruction the landslide caused. Here we have pholads, which are marine bivalves that bore into rocks below low tide, with lots of areas of pholads being exposed at our study site. Here's an old pillar covered in dead mussels. Mussels are also bivalves that touch the rocks in the low tide zone. They're left high and dry by the uplift. And another prime example are the bivalve cama, which are clams that dismember their substrate. For example, these large boulders. And amongst the cama are rock scallops, which are subtidal marine bivalves. From our results, there are numerous ways in which we can quantify this change. This graph here shows that the percent of coverage of living organisms in each quadrant. We are showing this trend for our most impacted site, our eastern section, with 2 different tidal zones, our mid and low tide, and are comparing it to our control values, which are our non-impacted areas. We see in January a decline from both tidal zones. This could possibly suggest the declining health of the ecosystem, or perhaps the winter storms in this already stressed environment. In the following months, we start to see an incline in coverage from both tidal zones, possibly suggesting improvement of the ecosystem. But this only tells us part of the story. We were curious to see which organisms were declining or recovering in our impacted areas. For this study, we want to focus on 4 major groups of organisms, which are color-coded as followed. Our sessile organisms, meaning they are unable to move, are in dark blue. Our grazers, which are able to move, are in dark green. Green algae, commonly known as sea lettuce, is in light green. And the rest of our algal types are in red. Now, typically in a healthy ecosystem, all of these groups would be noticeably present. But we see here in our most impacted area for the mid-time zone, between the months of October through December, all the algal groups present along with some sessile organisms. But we see a decline of everything in January. We now see the green algae become dominant over the following months. This could be because green algae is said to be an early colonizer of a stressed environment. And we see the same trend with our low tide zone of our high impacted site. All algal groups are present along with some sessile organisms for the first couple of months, everything declining in January, and with the following months being dominated by that green algae. We can see these changes expressed spatially along the Portuguese Bend shoreline. Here we plot the species richness, meaning the number of different species found within a specific area of our high, mid, and low tide zone of each transect. More species-rich transects have larger circles and darker blue shades, versus less species-rich are paler with smaller dots. We can see that our more impacted parts of the shoreline have lower diversity. Even though my project is focused on a few organisms, I want to show how much diversity is in the rocky intertidal. These beautiful organisms are constantly being exposed to changing tides and human interaction. Although this program is coming to an end, the landslide is still active, which means the changes will still be happening. We have documented the impact to the shoreline and the marine communities, but we're just now starting to see ecological recovery. It is hard to say when the diversity of the shoreline will recover and what that looks like, but I'm still very eager to continue conducting annual surveys with my mentors in the future. I would like to thank the museum for creating this program, NSF for funding it, and other individuals that have contributed to my project. Thank you.
Thank you so much, Chloe, for that awesome talk. So, any questions for Chloe?
Great talk, Chloe.
Thank you.
I was curious with some of the data that you have for the landslide, do you think you could make predictions of what the layout would be for like next year or the following years?
I would say it's hard to say because geology is so unpredictable. They do have stuff like water pipes to slow down the landslide, but since it's just a very unpredictable area, we're unsure what could happen next.
Okay.
We'll hopefully, with annual surveys, we'll be able to see that.
In terms of the, you said the sea hares?
Yes.
In terms of the research involving that, do you think that there will be a resurgence in those, or does this landslide effectively mean that they are no longer viable in that location?
Not necessarily. We are seeing some recovery of early organisms. For example, we see some snails being present, as well as some other algal groups. But typically, sea hares have a year life expectancy, so it was bound to happen. But hopefully, hopefully they'll recover soon.
All right, I think we are going to thank Chloe. Great job. There you go. We are now going to take a 10-minute intermission, and we invite you all. The second session is going to be done by Jason Gonzalez-Marie, and she's going to talk about human stomatolytes, investigating the role of biofilm forming bacteria in calcium phosphorus kidney stone formation.
So for my presentation, I'll be giving a talk about what project I've been going on for a couple years now. closely with the routinized and changed the way we do field biology, zoology, and the role of human microbiome as a whole. Before we jump in, I would like first to give a quick introduction to two parties that we believe hold strong significance in shifting the kidney stone pathogenesis, which is biomineralization. What is biomineralization? Biomineralization is a biology-induced process where living organisms, notably bacteria, facilitate the degradation, conversion, and production of inorganic minerals from organic substances. In other words, the process of microscopic living organisms produce minerals from organic material. There are many examples of this process in the natural world, and calcified cells and marine organisms, which are very long skeletal system where our bones are formed by osteocytic cells through ossification. However, I'd like to emphasize a specific example of this biological process, which are stromatolites. Stromatolites, one of the Earth's oldest non-living fossils, are low sedimentary formations that show that photosynthetic microorganisms known as cyanobacteria. In the photosynthetic development, these are bacterial colonies include biochemical cycles of the environment, and through adhesion, trap sediment will eventually solidify into mineralized microbial mass. The representation of stromatolites in context zoology is how we define our kidney stones. The kidney stones, or more specifically calcium-oxalate stones, is actually a bacterial in nature, meaning that they are not touched or characterized by the presence of bacteria. These cells make up 80% of all cases of kidney stones, and yet we still do not have the correct information for the underlying mechanisms that lead up to formation. However, when we control the internal structure of the calcium-oxalate stones with our stromatolites, we find out that they are remarkably similar. Both of they are ultimately layers of cells in like material, as we can know from evolution and geology, and also similar characteristics from only biologically mediated processes. These calcium stones are thought to be abiotic in origin. However, when we view these stones through a mineralogy lens, we see that is actually not the case. Instead, we think that bacteria is playing some sort of role within these stones due to structural similarities with stromatolites. So for our project, in collaboration with the Department of Urology at UCLA and the Mineral Sciences Lab at NHM, we want to figure out what is truly going on with kidney stone disease. So for our hypothesis, we believe that biofilm-formed bacteria play a causative and significant role in the formation of calcium oxalate kidney stones. To test our hypothesis, we must follow and address the following objectives: to visualize and identify microbial species in calcium-based stones, determine whether or not bacteria and biofilm are present within stone fragments, and evaluate whether stone properties, morphology, and chemical composition are a result of a biologically induced process. And to test our hypothesis, we utilized a diverse array of advanced imaging techniques to visually capture the internal structure of our calcium oxalate stones. Kidney stones were collected from human patients undergoing laser uroscopy at the UCLA Medical Center, and once we receive these kidney stones, we then prepare them for microscopy analysis under Raman spectroscopy, scanning electron microscopy, and fluorescence microscopy, and with a variety of biological staining agents. Staining the stones prior to microscopy helps us detect different types of bacterial components within our samples, including DNA groups, lipids, and extracellular polysaccharides. Initially, we want to characterize and identify the chemical composition of the binding seen within our stone samples. On the left, we have a polished calcium oxalate stone, and obviously we can see alternating layers of light and dark material. We want to figure out what these are. And with Raman spectroscopy, we see that calcium oxalate stones are not completely made of calcium oxalate crystals. The dark banding seen on the left are instead made of organic enriched material, not crystalline. Lighter regions correspond to high intensity calcium oxalate peaks, whereas darker regions correspond to low intensity peaks, which are characteristic of organic material. And when we plot up the data from our line scan, we get a graph like the one shown on the right, where we see an alternating, fluctuating pattern of crystalline organic material, similar to what we see in stromatic structure. However, the Raman alone cannot determine whether the organic enriched bands come from bacteria, so we must move on to the next technique. With scanning electron microscopy, you can actually get a higher resolution, super detailed image of the bacteria within the stone. After staining the stone fragments, we do in fact find structures that resemble bacteria within the calcium oxalate stones, which we thought were abiotic in origin. The tiny spherical blobs that we see here on the right zoomed in were actually identified bacteria, and we can see that these stones are completely filled with them, not only on the surface, but within its internal structure as well. And not only do we find bacteria, but we can also find other bacterial components, including biofilm. Biofilm, like we alluded before, is a slimy extracellular organic matrix that helps protect bacterial colonies in an extreme fluctuating environment. With the aid of false current coloring, we do not only get a glimpse of bacteria, but also the biofilm, which are color-coded in yellow. The biofilm exhibits a spongy, stream-like texture, and like the bacterial blobs, are found all over the place within these stones.
Well, these images are really interesting, but we still need to identify the bacteria within the actual bendings that we saw in the Raman spectroscopy. So as we move on to our final imaging technique, fluorescence microscopy. Following bacterial DNA staining, we found that these stone fragments have large areas of preferential staining, indicative of regions within the stone that are highly enriched with biofilm DNA. The images shown above will reveal well-defined striations of individual layers containing a high concentration of bacteria with high regularity in structure and separation from calcium oxide crystals. So the green layers represent bacterial biofilm DNA, whereas the darker red layers represent crystalline material. And all in all, we were able to gather enough evidence to suggest the possibility of an unanticipated parallel mode of kidney stone formation that is characterized by microbial activity. So what do we think is actually going on? Well, let's get through it. At the initial stage, bacterial colonization occurs within the kidney walls. These microcolonies start to secrete biofilm, possibly as a response of an environmental stressor. Biofilm secretion in the surrounding urine alters the local environment, which would then set up the conditions necessary for the biofilm matrix to act as a nucleation and template site for calcium oxalate crystallization. On the surface of these biofilm mats, crystals will start to precipitate and grow. Then new colonies of bacteria will start to attach themselves on the surface of pre-existing crystals, which will then be followed by biofilm secretion and crystal regenesis. Bacterial colonies will then be encapsulated within the internal structure of the growing stone, and the process repeats itself, resulting in altering layers of bacteria enriched in crystalline material. But the presence of bacteria alone cannot prove that our hypothesis is correct. We still need to unravel what are the actual mechanisms taking place to support this model. So for the last stage of this project, we want to replicate the possible growth mechanism that is resulting in the formation of calcium oxide stones, right? We want to see the actual process of biomineralization occurring in real time by observing changes in calcium oxide crystals when we introduce them to bacteria. So we're currently building a microfluidic kidney model to help replicate the urinary tract physiology in the lab. A motorized pump will inject calcium oxide crystals into a flow cell filled with bacteria and with a synthesized kidney-like environment. Within a synthesized kidney-like environment, we will be able to observe and document how bacteria influences crystal growth and morphology. Additionally, as for our future directions, we would like to identify the biofilm secretion pathways that are promoting crystallization. If we are able to discover what pathways are taking place, then we can develop targeted biofilm disrupting agents to help prevent kidney stones from forming in the first place. Kidney stone disease is still a prevalent issue in the modern world today. 10% of the world's population are impacted by kidney stones, and if left untreated, can become a serious life-threatening issue, especially for marginalized communities who lack access to proper medical care. Thus, it is extremely important for projects like this to exist. Integrating our knowledge of mineral sciences and geobiology with medicine can provide new advancements in the prevention and treatment of kidney stones, and interdisciplinary collaboration that can have significant impact on the way we address human diseases and improve the quality of life for many people around the world. And with that, I'd like to thank everyone who has supported me throughout my tenure as a researcher at the Natural History Museum. Special thanks to Dr. Aaron Celestine and Dr. Kamar Scotland for their mentorship and support throughout the year. Thank you to everyone here at NHM, including the cohort and the steering committee, as well as the research team back at UCLA. My buddy for emotional support as well. And none of this would have been possible without them, especially my buddy. And with that, thank you.
Thank you so much for that, Jason. I'd like to open up the floor for questions.
Hi, Jerson. Thank you. Are you noticing a similar crystalline structure of the kidney stones forming in every other kidney stone that you were using for this project, or is there a different structure between each?
Well, there's different types of kidney stones. We have struvite, calcium phosphate, calcium oxide. Even with calcium oxide, there's different types. There's calcium oxide monohydrate and dihydrate, which are different based on their chemical composition. But we do see it with struvite. So struvite, we do know they're infectious stone, meaning that they come from bacteria. They're usually caused by urinary tract infections, UTIs. And we actually use that to actually correlate to what we see with calcium oxide stones. Struvite, we're able to see the same bacterial blobs in the SCM, and we see the similar bacterial structures within the SCM as well. But it was difficult to do the ramen for struvite because it's pretty much organic, and we wouldn't be able to get like an actual characteristic peak for organics. Like I mentioned before, it's difficult to actually get a ramen spectroscopy for organics. That's why we correlated these low intensity peaks to organic material.
Thank you for that awesome talk. I loved the fact that you related them to the stromatolites. Can you tell me or us how that came about and how this whole concept came about? Because I just think this is absolutely fascinating.
Right. So I believe there's a student at USC who's working on stromatolites right now, and Dr. Aaron Stelzny, he studied these mineralogical processes as well. And we see that these are the processes that we see in other type of natural forming stones, but we do see, but stromatolites are like the one that I feel like everyone would love to hear about. So that's why I mentioned them before anything else.
All right, thank you so much, Jerson. Great job. Next, we're going to hear from Julian So on morphological shifts in western fence lizards across time and space. Thank you, Julian.
Hi, thank you for the introduction. Once again, my name is Julian So, and I will be presenting my research on morphological shifts in western fence lizards. Urbanization introduces new environmental conditions and changes, such as climate change, habitat loss and modification, and deforestation. All these are changes that can affect local plants and animals and place stressors on them to morphologically shift. But first, what is morphology? Morphology is defined as the form or structure of an organism. Some examples of this include the different shapes of leaves, the number of petals on a flower, and the different body sizes of animals. These are all examples of morphological traits that can change or shift in response to urbanization. So let's go over one example of this occurring. The peppered moths in 19th century Britain and Ireland are famous for their morphological shifts. Peppered moths are naturally white with black specks, such as this one here, allowing them to camouflage with light-colored lichens on dark tree trunks. However, in the 19th century, soot production from factories in urban areas released high amounts of carbon into the environment, which killed off the lichens and revealed dark-colored tree bark underneath. This allowed darker-colored moths, such as this one here, to camouflage more easily and evade predation, allowing these leading to the urban population of these moths evolving to be darker over time in comparison to their natural counterparts. So morphological change is affected by urbanization, influencing these moths' survivability and ability to pass on favorable traits to future generations. But let's look at this in the context of lizards. Prior research has shown that lizards from natural areas morphologically differ from lizards from urban areas. For example, one study by my advisors, Drs. Greg Polly and Bree Putman, found that the limb lengths and toe lengths of western fence lizards decrease with urbanization. Changes such as this are often correlated with how lizard populations respond to novel environmental conditions or the introduction of human-made surfaces, such as this lizard here on a slab of concrete. However, studies like the one I just mentioned mainly look at changes in urbanization across space and not through time, which leaves a gap in research for understanding how urbanization over time can impact morphology. So I wanted to examine this understudied relationship using western fence lizards. If you spent a lot of time in the western United States or the Los Angeles area, you likely have seen this lizard at some point. Western fence lizards are commonly called blue bellies because of their bright blue underside that makes them so distinct. They're found in a wide variety of habitats, including both urban and natural areas. And lizards in general have relatively low vigility, meaning that they don't have the ability to migrate or disperse as easily as large mammals or flying animals do. What this means for these lizards is that they are forced to respond to habitat changes and endure unfavorable conditions, making them ideal for studying the effects of urbanization across smaller scales. So through western fence lizard museum specimens, I wanted to address the following research objectives. Have fence lizards displayed morphological differences across space and time? And are these differences correlated with urbanization? To answer these questions, we first obtained around 250 western fence lizard specimens from the Natural History Museum of Los Angeles and the California Academy of Sciences. These lizards were collected from 6 sites across Los Angeles and Orange counties. We then classified each sample as historical, which meant it was collected between 1911 and 1964, or modern, which means it was collected between 2015 and 2018. We also determined whether each lizard was from a natural site or an urban site. Next, we collected data on a couple of morphological traits. The first is snout vent length, which is the lizard version of body size. We looked at head length, head width, forelimb length, fourth finger length, hind limb length, and fourth toe length. We also looked at a couple of scale count traits. We looked at dorsal scale count, which are the scales on their top side, ventral scale count, which are the scales on their bottom side, and fourth finger lamellae, which are the scales that run along their fourth finger. And finally, we analyzed the data using a programming language called R to detect any patterns in these traits. So I wanted to highlight a couple of key findings that we found. In modern lizards, body size is greater in natural areas than urban areas. In this box plot, the green box, which represents the body size of modern natural lizards, is higher than the purple box representing the body size of modern urban lizards. We also found that historical natural lizards were larger in various traits, the first of which is their head size. Historical natural lizards have longer heads than modern lizards. In this box plot, the orange box, representing the historical natural lizards, is higher up than the green and purple boxes representing the modern natural and modern urban lizards. If you look at the y-axis on this box plot, you might find it strange that there are both positive and negative values. The positive values represent head lengths that are longer than expected for a lizard of its body size, and the negative values represent head lengths that are shorter than expected for a lizard of its body size. And a zero value just means that the head length is exactly as expected. And you'll notice this weird y-axis on the following box plots as well. We also found that historical natural lizards have wider heads than modern natural lizards, and modern natural lizards have wider heads than modern urban lizards. Once again, the orange box representing historical natural lizards is higher than the green box representing modern natural lizards, and this box is higher than the purple box representing modern urban lizards. And we also found that historical natural lizards have longer hind limbs than modern lizards. Once again, the orange box representing historical natural lizards is higher than the green and purple boxes representing the modern lizards. So to summarize my findings, let's revisit our research objectives. Western fence lizards have displayed morphological differences across space and time. Historical natural lizards are larger in various traits than modern lizards, so it seems like over time, traits are shrinking in relation to the lizard's body size. And some of these differences are correlated with urbanization. Modern natural lizards are larger and have wider heads than modern urban lizards. So in short, we are detecting morphological shifts in some traits through time, and in other traits, they vary across space between urban and natural populations. So why does this research matter? My research looks at morphological changes through time, which is an understudied relationship. In this box plot that compares body sizes of lizards from different time periods, the orange box representing the historical natural lizards represented by the orange box and the modern natural lizards represented by the green box, they have significantly different body sizes despite little to no change in urbanization levels of their sites over time. It's possible that these lizards could be getting larger over time and something about urban areas could be limiting their change in size, but we can really only speculate because there just aren't enough studies that look at changes through time. And there needs to be more research examining this relationship to fully understand what factors play into the differences that we see here. My study also provides information towards understanding how Western fence lizards, a native species important to Los Angeles' local biodiversity, responds to human-driven habitat changes. The world is changing rapidly, and it's important to see if and how animals are able to keep up with these changes. Not all lizard species respond to urbanization in the same way. Studying more species allows us to meaningfully predict how lizards and other animals will respond to urbanization. And finally, I wanted to highlight the value of museum collections in studies such as mine and those of every other researcher presenting today. Museums serve as a library that gives us access to physical data from the past. Without museum collections, many long-term scientific studies would not be possible. I'd like to acknowledge all the people who made this project possible. My advisors, Drs. Greg Pauly and Bree Putman, the National Science Foundation, and the Natural History Museum of Los Angeles County for funding my research, the California Academy of Sciences for specimen loans, and Rachel Guttner, David Ledesma, and the UNLAB cohort and other NHM staff for their support. Thank you.
Thank you so much for that wonderful talk, Julian. Do we have any questions?
Great talk, Julian. Question about maybe one idea with why you're seeing the smaller body sizes. Do you think it's urban heat island effects? Because, I mean, it happens with sometimes birds, and also There was a study with Kristen Winchell's work in Puerto Rico looking at anoles there in urban environments that they got smaller in body size. Do you think urban heat island could be playing a role with why they're getting smaller over time?
I think that might be a role. People have kind of thrown around the theory that like a smaller body size would, or a larger body size would reduce the amount of evaporative water loss, or the effects of evaporative water loss, and that is definitely something that is affected by heat. I'm not totally sure, but that is just one theory that I've heard.
I'm curious about what the distinction between urban and natural are, because there's such a blend between the two, especially in Los Angeles County and Orange County. Where was your research being taken place, or was it just through the lab here at the Natural History Museum?
My research was mainly through the lab, but all these lizards were collected prior to me starting this project. The way that we distinguished between the urban and natural lizards was mainly through like impervious surface cover. So that's like whether they had like trees above them or things like concrete under them. Yeah.
Thank you so much, Julian. I think we're gonna move to the next topic. All right, next we have Naomi Rodriguez, who's going to talk about Who's Who, Marine Isopods, Enhancing Species Identification Through Integrative Taxonomy. Thanks, Naomi.
Thank you for that introduction. I'll be talking about marine isopods, and I'm a researcher at the Marine Biodiversity Center. So firstly, to start us off, I want to introduce you all to my friend, the isopod. So isopods are more commonly known as roly-polies, and you might have seen them hanging around in the garden, in heavily soiled areas, or just out and about on the sidewalk. They are a very diverse group of invertebrates, meaning that unlike you and I, they have no backbone. So some common misconceptions about isopods is that they are bugs or insects when they're actually crustaceans. So think more like crabs and lobsters. And some key characteristics include 7 pairs of legs, pleopods, also known as gills, and even terrestrial, meaning land-based isopods also have gills, which is why they like to live in moist environments. And also like crustaceans, they have an exoskeleton, which is an outer covering that offers protection to them. They also have incredible diversity in that they can live on land, in freshwater, and marine environments. And there are around 4,500 species in marine habitats alone. So this evening we'll be focusing on a few different kinds of marine isopods and the challenges surrounding who they are. For our specimens, we use the collections here at the Natural History Museum. Our historical specimens, marine isopods were so abundant in our collection, we were able to sample as far north as Canada, all the way down to San Diego. And we also supplemented new specimens from the Southern California Bight, which was arranged from Point Conception, California, all the way down to Punta Colón, Baja California. And just bringing it a little bit closer to home in our own backyards, we were also able to sample specimens from Redondo Beach, Ranchos Palos Verdes, and Point Fermin in San Pedro. And it was actually at this Point Fermin location where my team and I encountered 3 different marine isopod species, all within a very short distance of each other. And all 3 of these species are roughly around the same size, and if not slightly larger than the isopods you would see outside in the garden. So it's crucial for us to understand the biodiversity in our own backyards so that when habitats are changing, we know who is being affected by these changes. But despite their broad distribution and relatively large size, identification isn't always so straightforward. And this is often due to a lack of line drawings, photos, and SCMs in the original species description papers. So we're fixing the ID problem, and we're doing this by using integrative taxonomy. And this is a method that basically uses different forms of data to better understand biodiversity. In the context of our project, we're using DNA sequences as well as creating visual resources that focus on morphology. And morphology just means focusing on physical characteristics, So we're creating visual resources such as line drawings, stacked photos, and FTMs to supplement existing species descriptions. And I'll talk more about these in just a few minutes. To collect our molecular data, we pull tissue from specimens, often using a couple of legs, and then we extracted DNA focusing on 2 genes, 16S and CO1, which are both great genetic markers for letting us identify down to the species level. And lastly, successfully extracted DNA was sent for sequencing. So to show our results, we're presenting an unrooted tree showing sequence similarities for the CO1 gene, where the blue lines represent branches showing closely related groups, the colors relate to the taxa we identified, and each tip is one individual we sequenced. We also did this for the 16S gene, however, we got similar results, so we'll just be focusing on the 1 gene here. I know this can be a lot to look at, so we're going to break it down. So these 2 groups show sequence similarities between each individual. For the pink group, they are all relatively the same sequence, where there's no differentiation in their branch length. But in comparison to the blue group, there are slight sequence differences, with the length of their branches varying slightly. But from the big long branches you see, there can be many differences between the 2 groups that we identified as 2 different taxa. So again, just putting it all together in the larger context of the tree. We're also able to highlight that similar clustering supports that our morphological tools for identification are working. And to work conjointly with our molecular data, we dissected key features off of our specimens. We paired a camera with a microscope for photo stacking, and we used a scanning electron microscope so that we can take images of our specimens and magnify down to a level that we would not be able to with a normal microscope. And taking a closer look, in 1970, Miller and Lee described a new species of marine isopod with this 1 line drawing you see here on the left-hand side. And this is the only visual that we were provided with. So we hope that these images providing different views of the same species of the dorsal, ventral, and lateral views, as well as pairing them with SEMs, will be helpful in identifying specimens with more confidence. Another example of this was in 1857, when Stimson identified Pedentitia rusticata, again, with 1 line drawing. And this is based off of 1 individual. So we're missing a lot of the key features we would like to see because we're only provided with this 1 view here. So again, we're providing the dorsal, ventral, and lateral views to highlight those key features that we mentioned at the beginning of the talk. The exoskeleton, those 7 pairs of legs, as well as the pleopods or gills. And another way we've been putting morphology into practice is by describing a new species of marine isopod. And this species has always been around, but because identification is still so challenging, we're frequently stumbling across new species. And again, we're providing those ventral and dorsal views of the specimen. Another great tool that we've been using for identification is line drawings, as they are a biologist's interpretation of what's important to look at when you're trying to identify a species. So for our new species, we drew legs and we're highlighting the spikes on the legs as they are a diagnostic feature, meaning that they separate our new species from the original species. We've also gone ahead and drawn mouth parts as well as those pleopod or gill structures. And again, we're just pairing this with the SEM images that we took. So on the left-hand side, we provided images of the new species, and on the right-hand side, we've provided images of the original species. And we're just highlighting key characteristics that are different here. So their pleotelson or their butt ends are visibly different. So for the new species, it's more conical and more pointed, where in the original species, it's more rounded and bulbous. And I've also included these 2 photos at the bottom of the slide, just because I think they look really cool. They get to show us all that texture and all those smaller details that we don't normally get to see with the naked eye or with a regular microscope. So for our next steps, we hope to continue our genetic analysis by using genes such as 18S. We hope to collect more specimens from different localities to continue to add to our collections. And we hope to continue to photograph more specimens to show that amazing biodiversity within marine isopods. And we're doing all this in the hopes that isopods on the West Coast can be more readily identified by experts, so we know who's in our waters and how we can better protect them. And lastly, I want to acknowledge that all of this work would not have been possible without the support of all these amazing people here. Thank you to the Natural History Museum and the UNLAB Program, to everyone in the Marine Biodiversity Center who supported me through this amazing journey, to our colleagues at Cal State Long Beach, and our funding through the NSF and Paracarabid grants. Thank you.
Great presentation. What do you think makes isopods so diverse? Like, what is so special and unique that's in common in our chart, that we have in beaches at different spaces? Like, what stands out to you most about them?
Yeah, I think one really cool thing about them is that they've kind of evolved in so many different ways. They came from water onto land, which is why land isopods have gills. And they also just play so many different roles. For example, on land, they break down materials, so that's why they like to hang out in our compost. They really like to eat avocado seeds. And then also in marine environments, they play many functions. For example, they can also break down things. They can even be parasites that stick onto like fishes and kind of like live there. I just feel like they have so much to offer to our environment, and they also tell us so much about the current organisms in our environments and how if those different living things are missing, how does that affect how these isopods are also living in these environments? Thank you.
One more question out there.
One of the phylogenetic trees that you showed had all the sequences identical. Usually in populations you don't see that, or depending on the species that you study. Are there any reasons why you speculate to see that they're all identical in that one particular clade that you're showing?
Yeah, so the way that we used that tree, we used it more based off of the morphological tools we were using. So all the identifications we made on the tree were based on using keys as well as looking at our specimens at hand. So we were using the genetic sequences to kind of just confirm the identities that we had given to them. There were also parts in that tree that weren't completely as clean as those two groups. It's just more complicated in the sense that we still don't have those morphological tools that can give us that confident identification, which is why those genetic sequences are really important for us to start looking where we can fix identification.
Gracias por la presentación. Yo les voy a hablar un poco sobre lo que estuve trabajando este año en UNLAB y como parte del departamento de zoología. Para empezar, es importante definir algunos conceptos y el primero que tenemos es diversidad genética. En términos muy simples, podemos entender que la diversidad genética es la variación de genes dentro de una población o especie. Es decir, cuántas versiones tenemos de un gen que va a codificar para cierto carácter. En este ejemplo podemos ver que tenemos 3 ardillas, cada una estaría representando una población y podemos ver que tienen un arreglo genético único. Podemos ver que tienen una población con una sola variante, otra población con 2 variantes y la que está encerrada en rojo serían 3 variantes. Podemos decir que esta tercera población va a presentar mayor diversidad genética, por lo que podemos también suponer que puede responder mejor ante los cambios del medio. Ahora bien, la diversidad genética puede verse afectada por múltiples factores, pero para el propósito de este proyecto nosotros decidimos enfocarnos en el flujo génico. Y el flujo génico lo podemos entender como el intercambio de material genético entre poblaciones de la misma especie, es decir, el intercambio de estas variantes o versiones de los genes. Y esto sucede a través de el intercambio de individuos, es decir, un individuo de una población se mueve hacia la otra, se reproduce, diversificando su material genético. Al mismo tiempo, el flujo génico puede verse afectado por muchos factores. Uno de ellos puede ser la distancia, puede ser que estas poblaciones se encuentren muy alejadas unas de otras y esto impide que se muevan. También puede deberse a la habilidad de una especie para moverse, es decir, que hay especies que tienen rangos de dispersión muy amplios, pueden moverse kilómetros, pero también hay especies que van a moverse muy poco, incluso puede que no vayan más allá del lugar donde nacieron. Asimismo, también tenemos las conocidas como barreras potenciales de flujo génico, y normalmente son barreras geográficas, como lo son las cadenas montañosas, grandes cuerpos de agua como lagos, ríos y el océano, y también barreras creadas por el hombre, es decir, construir vecindarios, construir carreteras, vías de comunicación. Estas barreras consecuentemente van a provocar una fragmentación del hábitat, y esta es una imagen aérea que tenemos de Los Ángeles, donde podemos observar las áreas densamente urbanizadas a través de la luz que emiten, y también las áreas que podemos considerar como naturales, como lo son las montañas San Gabriel, Santa Mónica y Santa Ana. Podemos decir que lo que para nosotros los humanos representa una forma de comunicar a lugares que están lejanos unos de otros, desafortunadamente para mucha de la vida silvestre representa aislamiento y descontinuidad en el hábitat donde antes podía moverse con libertad. Teniendo esto en mente, nosotros queremos saber cómo es que la urbanización y la fragmentación del hábitat afecta a la diversidad genética y el flujo génico, y para eso decidimos escoger una especie nativa de Los Ángeles, conocida como Otospermophilos pchi o en su nombre común, ardilla terrestre californiana. Nosotros escogimos esta especie porque, además de que es nativa, tiene un rango de distribución amplio a lo largo de todo el estado de California y, por supuesto, en el condado de Los Ángeles. Además, es una especie generalista, es decir, que puede adaptarse con facilidad a distintos tipos de ecosistemas. Ahora bien, nosotros nos realizamos 3 preguntas principales para guiar esta investigación. La primera es identificar la estructura poblacional, es decir, cuántas poblaciones diferentes tenemos de la ardilla terrestre californiana aquí en el condado. También queremos saber qué tan diversa es genéticamente. Y por último, si existen barreras tanto naturales como del paisaje urbano que podrían estar afectando el flujo génico. Para esto realizamos varias cosas. Empezamos por la colecta de tejido fresco, de muestras aquí en el museo. Preparamos especímenes para añadirlos a la colección y después con las muestras realizamos secuenciación de genoma completo y realizamos distintos análisis como de historia demográfica, admixture, obviamente diversidad genética y flujo génico y algunos otros análisis estadísticos. Ahora bien, lo que obtuvimos o lo que esperábamos obtener era algo parecido a esto. Cada color representaría una población distinta, por lo tanto esperábamos obtener de 2 a 3 poblaciones, una para las Islas Catalina, una para las muestras de las montañas de San Gabriel y por último una para las muestras restantes del condado de Los Ángeles. Sin embargo, obtuvimos algo como esto. Todas tienen un mismo color, lo que representaría que todas pertenecen a la misma población. ¿Y por qué esto es sorprendente? Bueno, como mencioné antes, el flujo génico se puede ver interrumpido por todos estos factores, las montañas, las carreteras, el mar. Y nosotros tenemos todos esos factores, tenemos montañas, mar y tenemos carreteras. Sabemos que las ardillas no van a cruzar las montañas, obviamente no van a nadar grandes distancias solo para conquistar una nueva isla. Y la mayoría de nuestras muestras fueron de ejemplares que murieron intentando cruzar las carreteras. Entonces, ahora tenemos una nueva pregunta de cómo es que no hay una diferenciación. Decidimos después ver la diversidad genética y también podemos ver que la mayoría de los valores se encuentran en 0 o muy cercanos al 0. Esto puede sugerir que hay poca diversidad genética. Y en cuanto al flujo génico, normalmente se requieren de por lo menos 2 poblaciones para poder observar realmente qué está sucediendo. Nosotros solo obtuvimos una, por lo que podemos decir que no hay una barrera, o al menos no una barrera evidente de flujo génico. Ahora bien, la gran pregunta es ¿por qué? ¿Por qué no hay flujo génico y también por qué hay poca diversidad? Para esto realizamos un análisis de historia demográfica. Aquí podemos observar que el punto más bajo fue alrededor de 11.000 a 12.000 años durante el fin de durante el fin del Paleoceno. Lo que esto nos dice es que hubo una reducción dramática de la población y normalmente cuando esto sucede también hay una reducción dramática de la diversidad genética. Al mismo tiempo también podemos observar que este recuperó de forma muy rápida y que parece ser que se ha mantenido así desde entonces. Bien. También podemos decir que en términos evolutivos quizá no ha pasado el suficiente tiempo, necesita quizá más tiempo, o bien, y lo que probablemente nos está sucediendo es que los humanos están jugando un rol importante en el movimiento de estas ardillas, ya sea de forma intencional o no. Además también he sabido que les gusta subirse a los autos y terminar en lugares totalmente distintos. Una de nuestras preguntas, ¿cuántas poblaciones encontramos para el condado de Los Ángeles? Una sola población. ¿Qué tan diversa es genéticamente? Parece ser que tienen poca diversidad, pero para esto todavía necesitamos realizar más análisis. Y por último, si la urbanización o el paisaje natural está causando barreras de flujo génico, al menos nosotros no encontramos evidencia de ello. Ahora bien, ¿por qué es importante? Ya vimos que parece que están bastante bien nuestras ardillas, pero la diversidad genética resulta que es de donde las especies van a agarrar recursos y material para poder sobrevivir al medio ambiente, en especial ante el actual cambio climático. Entre más diversidad pueden ser más probabilidades de sobrevivir. Además, también estos estudios pueden ayudar a informar políticas y estrategias de manejo en la conservación que últimamente nos terminan como humanos beneficiando. Y para terminar, quiero dar gracias a todas las personas que hicieron posible este programa, a mis mentores, a la doctora Casey y la doctora Sora, a los demás mentores y, por supuesto, a los demás participantes de este programa. Gracias.
Hola, Clara, gracias por esta presentación. ¿Tú crees que haciendo otro análisis como esté usando especies de esta temporada sería los mismos resultados?
Diferentes. Question was, using the same analysis from species from today, other than using the collections, would you get the same results or a difference?
La mayoría de los animales que ocupamos son de años recientes. No incluimos muestras históricas como en otros proyectos que trabajaron, pero sería interesante saber si ha cambiado a lo largo del tiempo.
I was wondering if you'd considered expanding outside of Los Angeles County. Do you think you'd see the same, like, differences, or would you, at what point would you start to see another population, or any ideas about that?
There's another study that kind of did the same throughout, like, California, and at least for Southern California, they found no structure in the population. Only if you compare them to the Northern California squirrels, but mostly for Southern California, they are all like the same.
All right, thank you, Oksana.
Thank you.
Thank you so much for that talk. So next we have Shay Crowell, who'll be presenting Integrative Taxonomy of Native California Channel Island cactus snails. Thank you, Shane.
Alrighty, thank you, Allison, for that introduction. So before I dive into it, I wanted to start off by introducing the California Channel Islands, which have 8 islands in total, with 4 northern and 4 southern islands. Around 20,000 years ago, the sea level was significantly lower than it is today, and the northern Channel Islands were connected as a single landmass. However, the southern Channel Islands have always been separated. So my project is centered specifically on the southern Channel Islands, which include Santa Catalina, San Clemente, San Nicolas, and Santa Barbara. Islands are unique in that they can foster evolutionary changes and serve as important habitats for many organisms that are found nowhere else in the world. So for the past year, I've been studying land snails in the genus Xerarianta, which are also called cactus snails. And 4 out of the 5 Southern California species that are found in this group are only found on the Channel Islands. So the cactus snail species include the Catalina cactus snail, which is appropriately named because it's found on Santa Catalina, the plain cactus snail and wreath cactus snail, which are both found on San Clemente Island, and the bicolor cactus snail, which is found on both San Nicolas and Santa Barbara Islands. The speckled cactus snail is our only Southern California species in this group that does not live on the islands. Instead, it is found in San Diego with a range that extends into Mexico. So given their distribution across the islands, we had the following questions. The first being, how are the species related? And secondly, what is the relationship between the bicolor cactus snails on San Nicolas and Santa Barbara Island? And the reason we care about answering these questions is because it can further our understanding of species evolution, while also helping to inform conservation efforts, since we can't fully understand how to best protect a population that we don't know much about. This research also highlights the importance of museum collections in collaboration with other institutions to foster the advancement of science. We can answer these questions using integrative taxonomy, which is a multifaceted approach to determining the relationships between species. So for my project, I looked at both molecular and morphological traits, or in other words, the genetic makeup and physical characteristics of each species. So before I could get started, I had to find specimens, which was very challenging because this group is not very well represented in museum collections. So I began the search at local museums, including the Natural History Museum of Los Angeles County and the Santa Barbara Museum of Natural History. I then received loans from non-local institutions, including the Academy of Sciences and Cal Academy of Sciences. Later, we did fieldwork in San Diego to search for the speckled cactus snail, which is our mainland species, and the Palos Verdes Peninsula to search for a possible ancestral population of the Catalina cactus snail that is currently only found on Catalina. So once we had the specimens, I sampled a small part of their foot tissue. I then digested and extracted this tissue, and then went through a process called PCR, which stands for polymerase chain reaction. And this essentially is a process to increase the amount of DNA from the original sample. I then used gel electrophoresis to verify whether the PCR worked, and if everything went smoothly during PCR, these bright fluorescent bands should appear in the gel, which I have an example of on this slide. And also this is kind of false advertising because this often did not look like that, but it's okay. And these samples then were sent out for sequencing. So in total, I used 5 genes to sequence 5 parts of the mitochondrial and nuclear genome. Once we received our sequences, we analyzed them using the Mr. Bayes-Bayesian program through an online computer cluster to construct a tree of relatedness. This is also known as a phylogenetic tree. So this tree represents a portion of the 134 samples that I extracted, with the oldest successfully sequenced sample being from 1946. So to simplify what this tree means, I highlighted these 3 points in red, which represent a node or a common ancestor. Any species that shares a most recent common ancestor is more closely related to the other species. So this is kind of similar to a family tree. We generated many of these trees, but I've only included one for the sake of time. However, one important thing to note is that across our trees, the arrangement is generally consistent. So if we look at the tree, the speckled cactus snail is the most distantly related of these species. This could indicate that the ancestral species of cactus snails may have come from the mainland. Another thing to note is that the plain cactus snail from San Clemente and the bicolor cactus snail from both San Nicolas and Santa Barbara Islands are more closely related. And this grouping here indicates that the Catalina cactus snail from Catalina and wreath cactus snail from San Clemente are most closely related to each other and share a most recent common ancestor. But when we look at the different species of cactus snails that are found together on San Clemente Island, they are not as closely related to each other, which means that they came to the island as separate species and remained that way. This here is another tree with a few samples, which indicates that the bicolor cactus snails from San Nicolas and Santa Barbara are the same species. However, we would need more samples specifically from Santa Barbara to confirm this. So apart from molecular work, I also looked at a few different components of the snail's morphology, or again, their physical characteristics. And one of the ways I did this was by using scanning electron microscopy, or the SEM. And this allows us to get very detailed and up-close images. So I imaged shells from each species. However, it is difficult to tell whether certain parts of the shell can be diagnostic or used to identify the species based on the SEM images alone. I also dissected out each species' jaw, but the jaws seemed to vary based on the individuals and were not specific to the species. I then imaged their radulae, which are their feeding structures that are often referred to as rasping teeth, and these structures do not appear to differ majorly between species either. More recently, I've done many dissections to look at the reproductive morphology, which can sometimes be used to identify a land snail species, but I have not yet gotten the chance to analyze these, and I will be doing so in the future to see if they are different. So overall, we were able to determine the relationships between the different species of cactus snails for the first time ever, and we expect that the bicolor cactus snails on San Nicolas and Santa Barbara are the same species, but we currently have limited samples. The jaws and radulae do not appear to be diagnostic to species, and we will need to analyze reproductive morphology to see if it can be used to differentiate the snails. With that, I would like to thank my mentor, Jan Vendetti, my co-mentor, Marty, and everyone that helped me during the program, as well as NSF for funding my research. And thank you all for listening.
All right. Thank you so much. Do we have any questions for Shay? No questions.
What was
all the way up in the back?
Just want to know what was hunting for snails like.
So the only time we really hunted for snails or like solid life snails was in San Diego. And honestly, you just flip over a rock and they're there. So it's not very difficult. It's not like they're going to run away from you. They're very easy to grab. So, yeah. Thank you for your question.
Hi, great presentation. I believe you said that you found 2 different species on the San Clemente Island, and you found that through differences with PCR. I was wondering if you were able to identify differences in the morphological traits for both species as well.
So for the morphological traits, the only things that we've really analyzed yet are the jaws and radulae, and those didn't really seem to be good identifiers for between species. But I expect that the reproductive morphology would be different since they've been separate species for seemingly a long time. So.
Thank you, Shay. So that concludes our program. I hope you all are as impressed as I am with the quality of these amazing researcher talks. Let's get all the researchers to stand up one more time. Come on, researchers, stand up, stand up. Amazing job. You truly have accomplished so much in this year. It's been amazing to see you grow as scientists, as a cohort, and see you support each other through this whole process. Thank you in the audience for all of your support for our researchers and for this program.